What I would like to show you is the example that basically Davide already started showing you, what we have done together. Well, we work extensively together because we take most of our images with an Operetta CLS, which, as he explained, is an high throughput microscope. It can give you a lot of these beautiful images all in a session, so you can put a plate in and have a lot of image in one go. This brings the high throughput in, and then with the high content, we want to extract information about these images. What I'm showing here are endothelial cells. I don't know how familiar you are with these cells. They are at the inner lining of all blood vessels. We culture them in vitro, and we also obtain them from induced pluripotent stem cell. This one that you see here are UVEC, a very common model in the field. And the staining that you see here, okay, I also take for given that when I talk immunostaining, we are all on the same page. We are looking at endothelial cell junctions. So the orange thing is VA cadherin, which is a very, basically an exclusive lineage marker for endothelial cells. We are looking at the green. Nice dots are notch, activated notch, not the receptor, but what happens when you actually activated it. Other name for it is an ICD. And we know that when this thing goes into the nucleus, it starts being transcriptionally active. And then in blue, we have the DAPI or UXT or whatever nuclear marker you like to use. And this is done. So we use the nuclei for identifying the cells, as Davide has highlighted. And then we want to, our problem when we started with this was actually recognizing different junctions. And if you see, it's quite obvious to me, but I look at this image quite a lot. So I'll try to make it obvious for you. My finger is probably better. So if you look at this junction in the center here, looks like straight, linear, continuous. If you look at this just below, it looks a little bit more jagged and a little bit zigzaggy and stuff. If you look somewhere else in the image, not much in this, but in other there will be, you will find actual holes. So where there are no cells. When we talk about endothelial cells, this is something biologically relevant because endothelial cells needs to be closely juxtaposed to do their job. Otherwise, the blood, the interface between the blood vessel and the tissue is disrupted, and this is pro-inflammatory, pro-thrombotic, et cetera, et cetera, et cetera. So we are very interested in characterizing monolayer of endothelial cells because it's what I do mostly. And we are doing, in this case, we've done it this way. Obviously, this approach, since this is a workshop on image analysis, it's obvious that this approach can be used by any combination of staining. I'm making the example of endothelial cell throughout this 1 hour and a half, because it's the system I know best. But obviously, you have to apply to your own cell of interest. So in our cases, what we have done to show you a little bit how do we get to this nice PCA graph that you saw before, or the clustering. So how do we extract all this information? The simplest thing is that basically, you will notice here we are using, this is Columbus, the software that we are using here, and is an instance of Perkin-Elmer Harmony, or vice versa. Basically are the same software. One is running on a machine, one is running on a server. And Davide, correct me if I'm wrong. And they do mostly the same thing, which is giving you a sequential system, so you can basically add module to your pipeline and make the pipeline do something for you. The first thing you can actually do is find the nuclei. Very simple. It normally works out of the box because the DAPI is a very clear signal. When we go to the second part, I will basically spend, if you think is useful, some time on how do we distinguish what is relevant signal to what is not, and how do we really help this software to find the actual feature that we want to find. But first of all, let's see what is that we want to find. So the nuclei are quite simple. It works very easily out of the box. Let's say in this case we use this channel DAPI, and we use the method B. There are several in the software, and so this is quite straightforward. Second thing is when we look to the junctions, we go in a little bit more problematic field because in this case, we wanted to do two things with the junctions. The first thing was finding the cell, because as Davide have said, we have to find the cell before we can ask questions. We want to know, for example, the morphology of the cells. And now, right, since it's attached to a very powerful computer, they recognize automatically what they are seeing. Even if there are small discontinuities in the image, computer won't do that. Will easily do mistake in recognizing what is a cell and what is not. So we have to try, one of the things we are trying to do is teach the computer what we can do quite easily and natively. So find the cell. And then, as I told you before, one interesting question was characterize the continuity of this junction. In the previous work, we have basically counted the number of particles which pertains to each cell. So to make it, you will see later. For now, let's go on with find cells. Finding cell with this staining is not extremely straightforward, so we need some pre-filtering of the image. This is what you have done here, and basically take it as it is, and then perhaps we dwell a little bit more into the details. We have done 1, 2, 3 sequential filters to go to the level where we find the cell. Okay, so you see that before we were at step filter image 5, we need the filter 6 and filter 3 to actually being able to tell to the module find cytoplasm in Columbus to find the cytoplasm. And you see that in the red we have highlighted the junctions. This find cell is quite satisfactory. It finds cells, and they are quite the right shape. Obviously, endothelial cells tend to overlap, but very slightly. And so at certain point, you might question that perhaps these pertain more to this cell than this other. It's not really important when we go and explore a large number of cells, because the majority of the heavier characteristic will weight more on the general result. Once we found the cell, we wanted to characterize the junctions. So with one staining, we're already obtaining two information, which is interesting. And we have done this by filtering further the image and actually clustering, so subdividing the general junction structure that we see here in red. So as a generalized object, we basically found smaller object which pertains to the generalized junction object. And you see then that these objects are then a little bit more defined than the general one, since we can see that this is quite linear, this is quite linear, this is quite linear, and here we have a little bit of bits. In the work we have done previously, we simply, it's not really simple, but we did it, counted the number of these objects for each cell, which is methods that can be implemented in Columbus. It's not very complicated once you can find the object. And nothing. The number that we defined, we find out that were correlated to the continuity of the junction, which is what our eyes see by experience. And then we wanted to know, I told you before, that the green dots that you see in the initial image were activated notch. And despite, okay, this image is a little bit crowded, but it's interesting to see how we detect these features. We detected the little dots, and this is a decision you take before. You say you know your images, you know your biological data, and you know that basically notch can be in the nucleus, in the cytoplasm, the activated ones, or nowhere, simply because the cell is not active, notch active, let's say. In the previous work, we have decided to categorize the cell that we were finding in 3 categories, and I think these will be regarded as a positive cell because we have a lot of these green dots here and some in the nucleus as well. These will be regarded as negative because there are very few dots, and there are some somewhere where there are no dots in the cytoplasm and they're in the nucleus, but there are only in the cytoplasm. So we define this cell like this, and we create classes. All of this is done all within Columbus. So, or If you want to use cell profiler, can do similar things. Harmony can do similar things. So it's just a choice of what you do. The advantage, for example, to use Columbo or Harmony, if you work with Davide, is that everything is already in a huge data store that can be easily accessed. So in other contexts, you just have to have your big data storage connected to the computer you have the software that you play with. But anyway, once you have defined some characteristic, and you do it with these calculate properties modules here, which might be tell me how big are these dots, how intense the staining is, and what is the number per object. So relate the number of dots to the cell, specific object that is there, and tell me basically, for example, here I'm asking, tell me paint in green a cell that has more than 3 dots in the nucleus and less than 3 dots. All right, in the cytoplasm, I think. Well, anyway, you apply this rule, which are simple mathematics, simple logic, and you can basically classify yourself. So I think this might start answering your question about how you cluster your cells, so how you define cluster. And this is the thing we done later on in the same procedure. As Davide explained, we asked then, okay, we have said that the cell with certain characteristic is called cell A type A, or type B, or type C, and then we ask the software to tell us, okay, tell me if there are some cell type A that are clustered together, and we find this kind of maps, which is interesting. In the case of notch, it's very interesting biologically for endothelial cells, because it's known, it's been reported that notch in a monolayer is not or active or non-active, but is quite heterogeneously activated, and apparently in certain conditions it clusters, in certain other condition it doesn't, and we are trying to figure out what is meaning biologically. And this is the kind of questions that you can start answering with this kind of pipelines, and then obviously you have to go back to the bench, apply some perturbation to the system, reapply the image analysis pipeline, and see if something has changed, and if your hypotheses are predicted or not. The last step normally in this kind of analysis is telling the software that you're using, please, about all the numbers that we are using for understanding, for quantifying this or that. And this, you remember always, the computers only understand numbers. So they will always work on that. And you can basically list as many parameters as you want as a result. Some can be also the intermediate one. You just use it as a service to find certain characteristics, but perhaps you want to limit these to those parameters that are biologically meaningful to you. You filter them, you put them in a data set, and then perhaps you post-process with some data analysis technique. You can use software for multivariate data analysis and this kind of stuff to obtain principal component analysis, clustering, and other kind of stuff. Or more classical question like, if I give VEGF to the cells, how many dots are around? There might be less, there might be more. And this closed the first part. So this was an example just to introduce you to what I would like to try to do more interactively with you in the following hour or so. So if you have question now, for example, about this first slide, who can tell that, oh, come on, this is super familiar, you are really boring me. What is the histogram of the image? Why it is there? The type of images, really, I use ImageJ because it's very, very common and it's free, and you can download it now and use it, and there is for biologists, it's supported by the NIH, and is a very powerful software, and many colleagues, as you, have basically developed ways to actually do particular stuff. So how familiar are you with these concepts? So what is an 8-bit image? What is a 16-bit image? What is an RGB image? I go on? Okay. So this is the image. And this, I spot here, right here, an error, which is an import error, my mistake. I done this yesterday at 6. And it's telling me centimeters. It's just a scaling error, but just be aware of this, because if you tell me, okay, how big is this circle I paint here, will tell you centimeters. But this is just a notice. Despite the mistake, this is an image which has certain characteristics. It comes directly from Operetta, and this is a typical output of an image from Operetta. It is at 2,160 times the same, which is quite huge, and is a 16-bit image. So when we ask, what is an 8-bit? What is a 16-bit? Okay, when we talk grayscale, so one single color, gray because it goes to white to black, from black to white normally in case of fluorescence images, all the way the reverse on non-fluorescent image or, I don't know, phase contrast, stuff like that. But the main message is an 8-bit image can contain information that can be encoded in 8-bit. And since a computer understands zeros and one, you have to count 2, 4, 8, 16, 32, 64, 128, 256. Meaning that you can encode in this file 256 shades of gray, which is something that can distinguish, so you can distinguish between the gray 128 and the gray 130. If you encode the information in a 16-bit grayscale, you can count a little bit further, and you can get to 65,536 shades of gray, which means that you have a lot more information. Now, the important thing in image analysis is understanding when you need this information, because this is way heavier than this. And the computer will have problem with memory, in particular when you handle a bunch of these images. So here we come to the concept, when you really, really need some huge images, it's way better to do all of this in a facility, because normally this laptop won't be really able, or will take a lot of time handling huge number of huge images. But you can work with this easily if you need it. ImageJ has a startup. Why I'm telling you this? Because ImageJ, the first thing you can do, you can import an image and telling, okay, I don't like this 16-bit stuff. Let's turn in an 8-bit because it's lighter. This 8.9 megabyte of image will become easily a 2.5. And so it's much more manageable, but you're losing information. And second, even if it looks like harmless, because ImageJ does it for you and you don't see anything, what's going on the background is applying some algorithm, which might be actually really faithfully recapitulating your information, it might not. So the first consideration you have to make when you start with image analysis is know your images. Know exactly what it is that you are looking at, because this is fancy to see. I can play around with contrast. If we have time, yes, we have time, we will do it later. You can see much more or much less if you want, just by sliding on a software. Okay.
Do they have this image? Is there one image that we shared?
No, but I have everything here. We can, later on, I will play a little bit with ImageJ. Or if you want, while explaining, I can do it, but as you wish.
No, sorry.
Okay. The last thing, I think, out of this slide that I would like to say, another thing that is very important. When you work with fluorescence, in particular, take a very close look at what your image is. Because if you export from your software an RGB image, so the composite, the nice looking, like the first image I show you, nice orangey and green dots, etc., and you export as a single image, this might not contain all the information you need. For example, I can export in Harmony, in Operetta, a beautiful RGB image, the first that you saw. And this will be encoded in this code, which is a 32-bit RGB, which means that for each color in RGB space, this is okay. We don't have time today to go into the color spaces, but you can find a lot of information online. The simple message is that when you have a 32-bit image, which is huge, actually, because it has 32 bits, you have information at this level, so 256 levels for each color, and an alpha channel, which is here from the 25th bit onwards, another 8-bit of alpha channel, which represent the intensity. In this space, you can really represent a lot of colors. I think a few millions, 4 millions or something. But you have to think what is that you really are looking for, because if you're looking at single channel for the segmentation problem above, it's probably better a black and white image of 3 black and white images that are the 3 things that you are analyzing. And this is what Operetta does. It keeps everything separate and all maximal resolution. Last thing of this image, which is quite, of this panel, which is quite important to me, is the histogram. I will show you examples, but this is, you see your gray levels here. This is a binning of all your image. So all these 2000, okay, what comes from the multiplication of these numbers, all of these are binned in a single graph, and each intensity, obviously the height of the histogram represent how many events, how many pixels of that value are there, and the histogram so represent where is that your information state. So in image analysis, you really probably are looking for putting some line here and tell me what's positive, what's negative. And then this becomes a little bit trickier when, because again, your eye and the computer attached filter things also according to other algorithms, not only intensity. But the classic, the very classic of image analysis was threshold-based intensity. So you say, tell me what's positive, tell me what's negative. For the nuclei, works perfectly. For, I don't know, I have a proliferation marker like BRDU, EDU, KI67 with various protocols. These are very clear signal, or the cell is on or the cell is off. And this is easy to do. And these are the kind of things that Columbus, Harmony, out-of-the-box thing makes your life extremely easier. When you have some more complicated question, you have still, we can use this software, but we have to put a little bit more of thoughts. So, and this is really the end of the presentation. So from now on, I just made a list, and I will open image J and go on with the various bits, and you tell me what you